Good morning, everyone. So thank you for the introduction. My name is Britta. I'm a third-year PhD student from Ghent University. And today I will present the comparative whole metagenome analysis in lesional and non-lesional scalp areas of psoriasis patients versus healthy individuals. So psoriasis is an immune-mediated inflammatory disorder with a global prevalence of 2 to 3%. But its prevalence is highest in Western countries. It's a multifactorial disease, so it's influenced by lifestyle and environmental triggers, but also genetics play a role. The disease is not restricted to one particular body area, but at least 80% of the psoriasis patients suffer from scalp psoriasis or psoriasis capitis. And according to dermatologists, this is also the most difficult region to treat psoriasis. And as such, a role of the skin microbiome has been suggested. So we collected 83 top and back scalp samples from healthy individuals and 64 lesional and non-lesional samples from untreated psoriasis patients, after which we performed the DNA extractions, sent the samples out for shotgun metagenome sequencing, and when the raw data returned, I processed the data by trimming the adapter sequences, depleting the human reads from the microbial reads, and subsequently performing the taxonomic and functional profiling to get a better understanding of which microbial species are there and what they are doing. So after host depletion, we found that lesional scalp areas contained a significant lower number of microbial reads compared to top, back, and non-lesional scalp areas. And this was also already observed for other body areas affected by psoriasis, but also for other skin diseases. And so we also quantified the number of 16S copies using quantitative PCR, and here we also observed a significant lower number of 16S copies in lesional scalp areas. And this lower microbial biomass may be attributed to two factors. So first of all, lesional scalp areas have an augmented desquamation of human keratinocytes, which results in an increased background of human DNA. But on the other hand, lesional scalp areas also have a higher amount of antimicrobial peptides, which inhibit the presence of certain microbial species, and this also lowers the microbial biomass. Across the entire cohort, we found that the sebaceous scalp environment was mostly dominated by Cutibacterium and especially Cutibacterium acnes. However, we observed that lesional scalp areas were more dissimilar compared to top, back, and non-lesional scalp areas. And so we observed that lesional scalp areas contained less Cutibacterium species and an increased amount of Staphylococcus and other species. Then, for the functional profiling, we used human, and we looked at the differential abundant pathways using Maslin-2. And we found some significant altered pathways that involve key substances, including amino acids, carbohydrates, vitamins, lipids, fatty acids, and many more. And these functional alterations may disrupt skin homeostasis and activate immune cells. And some of these pathways that were enriched in lesional scalp areas are also known to be upregulated in response to oxidative stress, which is a known hallmark of psoriasis. Subsequently, we also looked at the virulence factor genes, and here we found some virulence factor genes that were enriched that encode for flagella, as well as antimicrobial activity, as well as for biofilm formation. And one of the virulence factor genes that was enriched encodes for the MTR-CDE efflux pump protein encoding gene, which specifically targets human antimicrobial peptides, including cathelicidin LL37, which is a human AMP that is known to be enriched in psoriasis lesions. So one hypothesis could be that some microbial species may be capable to evade a host immune response, dominate the scalp environment, and as such contribute to psoriasis pathogenesis. So in conclusion, we found that psoriasis lesions on the scalp contained a lower microbial biomass with a more dissimilar microbial community, and this involves fewer commensal species and an altered functional and virulence potential, suggesting that the microbial community plays an important role in psoriasis capitis and generating new hypotheses to test. And with this, I would like to end my presentation. I would still like to thank my promoters and the collaborators from CMET, as well as my funding source, the FWO, and I would like to thank you all for listening. And if there is time for questions, I'm happy to take some.
Thank you very much. Maybe we could take any short questions.
It's a bit paradoxical. It sounds a bit paradoxical. I would have expected more bacteria actually than less.
Yes, yes, we've seen less, and other studies also found less bacteria and fungi. And so we also normalized against the ACTB gene, which is a human gene, and then the difference was even more significant, that there was a lower microbial biomass. But yeah, I think with the explanation of the fact that there is more desquamation of human keratinocytes and an increased amount of antimicrobial peptides, I think that could be a good reason for having a lower microbial biomass.